welcome to Sprechen Sie Science. Thanks for joining us today. We're going to be discussing Hardy Weinberg Equilibrium. So the Hardy Weinberg formula is used to predict what the population frequencies would be like if there is no evolutionary change taking place. So it's used as a null hypothesis. And since it is used as a null hypothesis, there's some assumptions that need to go with it because it doesn't really happen in the environment. It can only be replicated in the lab. So you can only have a population that's in equilibrium in a laboratory. So first thing you want to have is that you want to have no mutations, meaning the sequences are not changing, there's no new genes being created.
And there's nothing around that could cause the genes to change. No, radioactivity or, randomness.
So you do not want any change in your genes. You don't want new genes. You want to stick to your old genes.
You also don't want members of your population to migrate because that'll just change the pool of available genes.
So you don't want anybody leaving and you don't want anybody coming in because they both mess up with your frequency.
You don't want anybody being selected for because it also leaves you with some more of some genes and some less of some other genes.
So mostly you want everybody to have the same chance at survival. There's some fitness.
At least random mating at least should keep you from recombining any genes to make other combinations that could fall under any of the other forces.
Right. Also, if there is random mating, it's referring to a type of selection, which is sexual selection, which would be also driving the frequency of one or the other allele up, whatever phenotype is preferred. So no selection. You just find someone from the opposite sex and have offspring.
And then you also want your population to be as big as possible, because in smaller populations, the effects of any of the other evolutionary forces will be felt a lot faster.
Also keep in mind that this is a statistical model, so the bigger your population size is, the more it will follow the projections.
All right, here are the equations.
Okay, so as you know, it's the binomial expansion. So p plus q squared gives you your regular formula that you're actually using to get your genotypic frequencies. And also remember that it's all supposed to add up to 1.
Or as close to 1 as possible.
As close to 1 as you can get it. Okay, so it is really, really important when you're working with these formulas that you know what each of the variables stand for. It will save you a lot of trouble and a lot of confusion if you know exactly what each variable stands for and you don't get confused between the frequency of the dominant allele and the frequency of the dominant genotype. So the first thing we want to do before going into examples is defining each of the variables.
So P is going to always be the frequency of your dominant allele. Q is always going to be what you use for your recessive allele.
Then you have P squared is always going to be the frequency of the homozygous dominant genotype. And 2PQ is the frequency of the heterozygous genotype. And Q squared is the frequency of the homozygous recessive genotype. Now, each of these sets have a name. So these are the allelic frequencies. Notice that they refer only to one allele, either the dominant allele or the recessive allele. And these are
the genotypic frequencies.
And they refer to each of the three specific genotypes that you can have. So homozygous dominant, heterozygous, or homozygous recessive. So here's a sample problem, and we're going to give you guys a little bit of time to read the problem and figure out what you need to do.
So in our situation, we have 237 mice, and 89 of them are tan. So they stand out to you because they are the recessive group. And from these two quantities, we're going to come up with the allelic frequencies and the genotypic frequencies.
Okay, so remember from before that we said that Q is going to stand for the frequency of the recessive allele, and Q squared is going to stand for the frequency of the homozygous recessive genotype. So which of these two can you calculate with the information that you have just now? You have the population size, and you have how many of them are tan, which means how many of them are homozygous recessive. So you can right away get Q squared, and you just divide the total number that are homozygous recessive by the total population size, and it gives you 0.38.
Right, where it's a genotypic frequency, which is just a proportion of either dominant or recessive out of the population.
Right? And then you actually calculate Q, which is your frequency for only the recessive allele. And for that, you just take the square root of what you got for Q squared. And that gives us 0.61. Now that we have Q and Q squared, and I'm going to keep putting them up here in the corner what we already have, let's calculate P. So don't forget that P plus Q is equals 1, remember. All the three formulas all equals to 1, so that is very, very useful for you. And then we just subtract Q from 1, and you get 0.39 for P. Okay, so now that we've calculated P and Q, we just need to get everything else in the formula, and recall that we also have Q squared from the very first step, so we're just missing two terms, which is P squared, and you just square P for that, and it'll give you 0.15.
And then you're also going to just need 2PQ.
And you just substitute P and Q in, and you get 0.48. And the very last step, since we have everything that we needed, is to check your work. How do you check your work? Well, remember that this is supposed to add up to 1, so you just add them, and they're equal to 1.01, which is pretty close to 1, right? Would you take that?
I'll take it.
So you're done.
Look at
Perfect.
that. You get a check mark.
Yes. That's the college version of a star in your notebook.
Good job, sticker.